Okay, so for those of you that don't know, the UK Cereal Pathogen Virulence Survey was established in 1967 following an outbreak of yellow rust on the previous resistant variety Rothwell perdix, and the survey aims to identify changes in pathogen populations and detect new races that may have adverse effects on cereal production in the UK. And we rely on yourselves, so farmers, agronomists, trial staff, breeders, researchers, to send in infected leaf samples when they see them out in the field. And we're currently predominantly funded by AHDB, by the levy payers, but we also do have a proportion of funding that comes from AFA. And the two pathogens that we surveyed in 2023, as John mentioned, are yellow rust and brown rust. So for those of you that don't know, when a sample is received at NIAB, Amelia will incubate the samples and multiply each of the isolates that's sent in. And we also store an infected leaf segment for genotyping. We then, out of the samples that are sent in through the year, we whittle down to just 25 isolates per pathogen that we're able to test on our differential seedling screens. I'm going to talk a little bit more and explain about those on the next slide. And then we use that data and select 5 isolates for yellow rust and 5 isolates for brown rust that we do further testing on, looking at the adult plant reaction and also the seedling reaction for all of the RL varieties and candidates. And we put all of the isolates into long-term storage. So we have an excellent collection of all of the pathogens that we've surveyed across the years. So the seedling differential test, these are carried out in these modules that are shown in the top right-hand side of the slide. Each module has a different accession, wheat accession, which we know has a certain resistance gene. So an example is in the table here. So these are the different accessions, and then these are the corresponding resistance genes that they carry. And we will inoculate one of those modules with an isolate of yellow rust or brown rust, and then we'll be able to see which of the accessions the isolate was able to cause disease on. And then we can build up this the pathotype of each of those isolates. So this is its virulence profile, and this basically just lists the virulence genes that the isolate carries. So this is where you'll see in our annual reports, or when we mention that sort of long list of numbers and some of the additional cultivars. So this is just a description of all of the accessions that isolate causes disease on. And that way we're able to characterise the isolates, see which ones have the same pathotype or whether we have something new. And then we use the pathotyping information, and we have this race naming system, which in the UK we use different colours. And so this is just an example of how you decode the naming system. So there's WYR for wheat yellow rust. Then we have the different colour for the different groups. So predominantly in the UK we have the red group present, but we also have some from the pink and the purple group. So these are different races of yellow rust. And then the number is allocated sequentially. So and that relates to the pathotype that we see at the bottom. So when we get a new combination of those numbers and cultivars, then it will get a different number. And the pipeline that we've been following for the past few years is we receive the samples in the spring, summer. Occasionally we do get some from the winter. And Amelia is busy reviving and bulking these isolates. And then, as I mentioned, we select 25 of these isolates and we do the differential seedling screens in the growth rooms. And this is usually carried out in the autumn of that year. Then we select the 5 isolates that we screen on the RL varieties and candidates. And this is carried out, or has been carried out, in the spring of the following year. And this information is now a separate line in the RL booklet, which gives you the young plant rating. And then we have been doing adult plant trials on all the RL varieties and candidates, and that's performed in the following summer to when the isolates were collected, and that's used to validate the watch lists. So I'm just going to hand over to my colleague Amelia, who's going to run through this season's data. Thank you.
Good morning, everyone. Nice to see you all here today. I'll be taking you through the wheat yellow rust first. So I'm going to start today by talking about the adult plant trials, and these are the adult plant trials that we ran in 2023. And there are 5 individual trials for yellow rust, so we inoculate them with 5 individual isolates. And this is the isolates that we chose for those inoculations. So you can see we selected 22038, that one came from Costello and from Norfolk, and it was selected because it had this pathotype that was first seen in 2021 and then it reappeared in 2022. We selected this one here, 22055, from Devon and Gleam. That one was selected because it had this quite simple pathotype. We also selected 22143, another one from Norfolk, and that's because we had some quite interesting samples coming in from Norfolk in 2022. And they didn't come from the same trial. The Costello one came up from near the Wash, and the Mayflower one came from much closer to Norwich. And this isolate here had this pathotype. It was another new pathotype. It was A virulent on YR4, A virulent on Rendezvous, and A virulent on Warrior. We also selected this one, 22162, which came from Lincolnshire, and it was selected because it was genotyped as a pink isolate, and we don't see many pink isolates each year. I do have some slides on those later in the programme. And it also infected the additional cultivar Crusoe at seedling stage. And we selected 22180 from Theodore, and that one was selected because it actually carried the most common pathotype in 2022, belonging to Red 26. So I'm now going to show you just some of the results from the adult plant trials. I've expressed this in terms of mean percentage plot infection over 6 assessments. So I started my assessing on the 18th of May at growth stage 39, and I continued to the 21st of June at growth stage 71. Normally I would have tried for another assessment after that, but 2023 was quite dry and warm and sunny in June, and I found the yellow rust went over by the end of June, so I stopped assessing at growth stage 71. So each bar on this chart represents a different isolate. So we have 22038 in blue, 22055 in green, and so on. And it's a mean percentage plot infection from 2 reps. So the actual final scores that these varieties would have scored at the end of the season at growth stage 71 would have been much higher than these percentages shown here. But I just wanted to point out this is a calculated means. So we'll start with Robigus. So that's one of our susceptible control varieties, and it had the highest level of infection with the isolate in green, which is 22055, with 43.6%, just a smidge more than 22038. The next variety I've shown is KWS Zayat. That one was rated 3 last year, and it was our most susceptible RL variety in trials. Then we have Skyfall, also rated 3. You can see it was slightly more susceptible to the isolate 22162 in pink, but it was infected in all 5 trials. Then we have SY and Sitor and Gleam with a rating of 5. So you can see we have these big peaks here with the isolate 22055. And if you remember from the slide before, it was 22055 was originally sampled from Gleam, and it gave the highest levels of infection on Gleam and Inciter. And also I put up RGT Wolverine there with a rating of 4, which had between 5 and 15% with all the isolates. So next I wanted to show you the varieties that were rated 7 or more. You can see there's been a change on the y-axis here. If we go along the top line first, we're starting with LG Skyscraper, which had a percentage mean of 3.4% with the pink isolate, 22162. RGT Wilkinson, another variety rated 7. It was actually only susceptible to just 2 of the isolates, 22038 and 22162. RGT Illustrious, rated 8 last year. That one was actually resistant to the pink isolate. We have Cadoceilum there, so that's our most susceptible variety rated 9. And you can see it's got this green bar for the isolate 22055. And it was interesting to note, it remained really clean all season. And it wasn't until the last couple of assessments that it was just showing infection with one plot of that 22055 trial. Last on the row we have RGT Barstow with a rating of 8, again slightly more infection with the pink isolate. So if we move on to the bottom charts now, again we've had to change the y-axes again just so we can illustrate them to you better on the slides. So RGT Stokes and LG Redwold, both rated 7. You can see they showed a very similar pattern with Stokes having slightly more than LG Redwold. We got RGT Rashid in the middle there with a rating of 8. Again, showed more infection with 2 of the isolates. LG Illuminate rated 7. That one was showing more infection with the 22038. But you know, these are quite trace amounts. If you look at the scale, it's just over 0.6% there. And lastly, Graham rated 8. And that one was also showing in slightly higher infection levels with the 22055 isolate in green. So I wanted to have a look at our top 5 most widely grown varieties. And I've used the weights certified for 2023 to illustrate this. So Cadoceilum dorsum with a rating of 9, that was the most widely grown, most popular variety in 2023. You can see it's it stood up to its rating of 9 and performed very well. Whereas Cadoceilum extaze rated 8, and that was our most susceptible rated 8 variety. And you can see it did actually have this higher level of infection with the pink isolate, 22162, and is what I would have expected from that isolate. Champion rated 8 was resistant to all 5 isolates. We have Skyfall there, so it's the 4th most popular one. It's rated 3, as we've seen. It is susceptible to yellow rust, but it's still a popular Group 1 milling wheat. And Crusoe, finally, rated 9. That one was also resistant to all 5 isolates. I think it had a smidge there with maybe 22038, but it's performed very well in our trials. I was expecting to see some infection on Crusoe with my 22162 isolate because it did infect Crusoe at seedling stage, but it didn't translate at the adult plant stage. So if we have a look at the isolates that were resistant to all 5 isolates, I'm going to exclude the candidates on this slide because I'm going to talk about those on the next slide. And these are the ones that showed 0.0 over those 6 assessments. So completely clean, no pustules, no stripes, and they all performed really well. You can see we have 3 varieties rated 8, which are Champion, LG Prince and Merit, and all the rest were rated 9. So they did well. And you can see Costello and Mayflower, they're up there, so they were resistant. That means those 2 isolates that came from Costello and Mayflower, which were 22038 and 22143, they didn't manage to infect their host varieties at adult plant stage. If I've missed out any of the RL varieties, you can find our full results in the 2023 UKCPVS annual report, and that's already on the AHDB website for you to have a look at. So the candidates, how did they perform? So you can see I've just highlighted some of the reactions here. So LG partridge, that was our most susceptible candidate, having infection in all of the with all of the isolates. If we just have a look at the ones that scored 0, 0 throughout those 6 assessments, you can see we have KWS Dragon, LG Grendel, LG Arkle, KWS Scatum, Blackstone and Belinda were scored 0, 0, so they did very well. And then I just thought we'd have a look at the ones that have actually stayed on the recommended list for this year. So we've got SY Cheer, Bamford, Almara, Blackstone and LG Beowulf. They all did really well. You can see a little bit of yellow highlighting, but that's just trace amounts. They all performed well in our trials. So my take-home messages for the wheat yellow rust adult plant trials. So the most susceptible RL variety in trials was KWS Sciat. KWS Sealum with a rating of 9, that was our most susceptible variety, rated 9. It did see some infection in one trial by the end of the season. KWS Dorsum, another rated 9 variety, the most widely grown variety. That one was resistant. It did have a little hint of yellow rust with 22038, which you probably couldn't quite see on that slide, but basically it did perform very, very well. And we can say that many of the RL and the RL candidates were resistant to all 5 isolates. And the candidates that have been added to the recommended list for the 2024-25 season performed well in our trials. So we're going to move away from the adult plant trials and now focus on the samples that I received into the UKCPVS in 2023. So I received 92 samples. That's a little bit down on previous years, but we did find that the weather was not very conducive to rust development, and it seemed to be more of a septoria year out there, with a lot of natural infection trials not really seeing that higher level. So my 92 samples, they came from 21 counties. We got quite a good spread across the country. We've got samples coming in from the south, the central and the eastern areas. We've got samples in from Yorkshire, also Tyne and Wear. Scotland was well represented, and we also had samples in from Northern Ireland. So those 92 samples came from 37 different varieties. And it was KWS X-Days rated 8. That one was our most sampled variety. So we had 13 out of 92 samples in from X-Days. Skyfall was our second most sampled variety with 10 out of those 92. And then Graham at the bottom of the pie chart, that one had 8 out of 92 samples. So you can see we had a good range of samples from different varieties in different counties in 2023. So we selected 36 isolates for a seedling differential test. I know Charlotte's just been mentioning that we only select 25, but that's because we are funded to test 50 rust isolates altogether, and I didn't get that many brown rust samples in. I will be talking about those next, but I ended up testing 36 yellow rust samples to make it up to that 50. And these isolates were selected based on their host variety, the location, any interesting notes that came in with the sample, and we also considered the percentage area grown of the host. So I'm not going to go through all of that table there. I just wanted to pop the information up just in case you wanted to have a look to see if your sample was tested. Every time I receive a sample into the UKCPVS, I do email the sampler back and say, oh yeah, thank you very much for the sample. Your sample's been allocated the isolate code of 2023, blah, 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 blah. So you can see these are the ones that we selected for testing. So we selected from a really good range of host varieties. We've got susceptible varieties like Zaya in there, like Skyfall. We've got intermediate varieties like Gleam. And we've also got resistant varieties like LG Astronomer. We've even got LG Typhoon in there. That's a name I don't say very often when I'm talking about yellow rust. And we also had select them from across the country. So I've got ones from Lincolnshire, from Fife and Midlothian, Cambridgeshire, and also Dorset. So we just try and spread out our net as wide as possible when selecting those samples. I did want to point out the last 3 isolates on the list, 23, 86, 89 and 92. They all came from Midlothian, but that's because AHDB alerted us to the fact that they were seeing some unusual levels of disease in a particular RL trial up in Midlothian. So they asked the trial manager up there to send us some samples, and they all came in late in the season, and we selected 3 from that trial to have a look at. So I'm going to express my yellow rust seedling tests in terms of the percentage of isolates carrying virulence. So this is the virulence for the YR genes, those differentials that we included in the seedling tests. And I've shown you the data for the last 5 years. So as you can see, the main resistance genes were found in 100% of the population. In 2023, we had a slight decrease for YR3 and YR4. But the one that we always keep an eye on is this one here, which is virulence for YR8. You can see that over the years it has been seen in some of our isolates. It wasn't detected in 2022, but we did see it in 3% of samples in 2023. And no virulence was detected for YR5, YR10, YR15 and YR24. So looking at that, you think, well, there hasn't been that much change in the population, but it's not until we actually look at our additional cultivars that we can see much more variation in the isolates that we test. So, as we've been talking about the pathotypes, these are the additional cultivars. So when you see the pathotype and it says RE, that means it's rendezvous, WA is warrior, sterling is ST, and we also have Chronic, Apache, Evolution and Crusoe. So we have lots of fluctuation over the years, but in 2023, virulence for Rendezvous, Warrior, Cabo Sterling, Apache and Evolution all decreased, and, gone the wrong way, virulence for Chronic and Crusoe both increased. And in fact, Crusoe was seen in about 22% of the isolates that we tested. So the pathotypes that we found in 2023. So the most common pathotype belonged to this the red genetic group, and it was seen at 14% frequency. We had another common pathotype. You can see I've put red or pink. That's because we had a group of isolates that all showed that pathotype, but we genotyped some of them. One of them fell into the pink group and the others fell into the red group. And that's not unusual. We have seen that before, where the pathotype that we see in our seedling tests can fall into 2 different genetic groups. Did we detect any new pathotypes in 2023? We did. We have this top one, which had virulence for YR8. That one was not classified purely because we didn't genotype that particular isolate. And then we have these other ones. We had one belonging in the red group. Purple, again, I've had to put 3 or 6% frequency because I had 2 isolates that carried that pathotype. One was genotyped to be in the purple group, the other one we didn't genotype. So it could be a purple group, or it could be probably a red group. We'd need the genotyping to confirm that. And the last pathotype on there, new for 2023, was in the red group, and it carried this very simple pathotype. It was avirulent on a YR3 and YR4, and didn't infect any of the additional cultivars. So to try and bring you information quicker and improve the relevance of our the information that we can bring you, as well as doing the normal seedling test, we piloted an approach to actually include more RL varieties in our initial screen. So these varieties along the top there were included in those differential tests that I did on the full 36 isolates. So we're calling this our RL subset. And me and Charlotte, we had to select which RL varieties to include. It was really quite difficult. I had a limited the way we set our tests out, I had a limited number of spaces, so I couldn't include all the RL varieties in this particular screen. But I had a look, and we were looking at ones which we thought would produce the most valuable resistance data, and how they'd previously performed in our seedling test the year before. So these are the ones I picked out. And you can see this almost checkerboard of R and S's. So this is their reaction at seedling stage. So if they were susceptible at seedling stage, they have an S. If they were resistant at seedling stage in my screen, they have an R. And it highlights the diversity of the yellow rust population, because we have such a wide range of reactions. But we can say that 83% of those 36 isolates infected Graham, which is rated 8. And 22% infected Crusoe and Ixtase. 8% infected Xylem. So we know Xylem, it's got an adult plant resistance rating of 9, but it was susceptible in three of the isolates, and those 3 isolates came from that trial in Midlothian that AHDB alerted us to. And 3% infected Merit, RGT Barstow and LG Illuminate. So 3%, that's just one isolate, but it was quite interesting that that one isolate was able to overcome the resistance of those 3 RL varieties. And that isolate was genotyped as red. And lastly, swallow, it was resistant to all 36 isolates. I know swallow's now dropped off the recommended list for this year, but I always used to keep an eye on it because it always performed pretty well in my adult plant trials, and it's always resistant in my seedling tests, despite its moderate disease, adult plant disease rating of 6. And if you want any more information on this RL subset, AHDB have a really useful publication that they put out in October, just to explain these tests in more detail, and you can have a look at that and just have a look at the results on that bulletin. So I wanted to explain how the UKCPVS feed into the RL. So the data from UKCPVS is now included in as this yellow rust young plant data, and it's on a separate line in the RL booklet. And just point out the link there as well to the AHDB website. They've got a nice explanation of the young plant resistance and how it's worked out. But my next slide shows it in a little bit more detail. So, as well as this pilot screen that I did with the 36 isolates, we've chosen our 5 isolates to test on the full set of RL and RL candidates. And these are my 5 isolates here. So you can see, if it's an R, it was resistant at seedling stage, S is susceptible at seedling stage. And the young plant data is generated from this seedling data with these 5 isolates. And it also takes into account RL trial data before ear emergence. So basically what that means is if AHDB noticed that a variety that I'm saying is resistant in my seedling test, but they're seeing infection on field trials, they can change that rating to an S if they feel it's appropriate. So this is a little sort of sneak peek really at how the R and S will look in the future, because the current booklet that's just been produced has the information from last year's isolates. So this is hot off the press. I literally did these tests in December, and it's not published information yet. But basically, anything that was susceptible to just one of these 5 isolates will show as an S on that young plant rating, and if no isolates overcome the resistance, it will show as an R. And we can say that varieties that are susceptible to multiple isolates are likely to be liable to problems in the field at the young plant stage and should be a priority for monitoring. That's purely because there's more isolates out there that can affect those varieties, so you just need to keep an eye on them. Now, you may be thinking, if I've tested the whole set of the RL and the RL candidates, why am I just showing you a small selection of them there? Well, I have them all on the next slide. So these are my most recent results. Anything that was susceptible to one or more of those 5 isolates is in my yellow box, and anything that was resistant to all 5 isolates is in my green box. So we've got the new candidates up there for 2024. And you can see, we've got some surprising ones in there. So the Zealam's in there, RGT Rashid is in there. We have some of the candidate varieties, but we actually have quite a lot in the green box for yellow rust. That they were resistant in my seedling tests. So it just shows that the breeders are doing a really good job out there, and they're helping to produce resistant varieties. So I've just got a few slides on genotyping of our wheat yellow rust. So the aim of this is to conduct routine genotyping of wheat yellow rust isolates using results from Diane Saunders' JIC Field Pathogenomics project, and following the Marple pipeline, which categorises isolates into the different genetic groups. So we've been doing this for 5 years now, and you may be thinking why we're only, genotyping 24, and that's just because the way the test is set out, we can do multiples of 24, and it's quite an expensive process. So 1 year we did manage to genotype 48 isolates, but we usually just stick to 24. In 2023, we did try genotyping 24 isolates, but one failed, so I've only got the results for 23 of them. And the genotyping at NIAB is done by my colleagues Helen Bates and Anne Webb. So I'd just like to thank those two colleagues for their work in this, because I don't do the genotyping, they do it for me. So we can say, as a roundup, that the majority of the UK genotyped isolates from 2019 to 2023 belong to the red group. So we have this big phylogenetic tree here. I won't go into too much detail because it's too big to show you in detail about it, but basically the genetic groups are in these different colours. So the Marple genotyping can separate the isolates into those different groups. So the red group, you can see that's dominated the population for the last 5 years, and we don't just have our isolates in here, we have also reference isolates that Diane included in her work, the results of those in there. Round the outside, with the sort of like yellow and sort of like brown blocks there, that actually showed the results from our seedling virulence tests, our differential tests. But if we focus on the central section, that's the genotypic groups. And in 2023, when we genotyped 23 isolates, 21 belonged in the red group. And we found 1 isolate in the pink group and 1 in the purple group. So in the top right-hand quadrant, we've got the pink and the purple groups. and you can see that we don't detect that many isolates in those two groups over the years. I think in the purple group we had an isolate in 2019 that was purple, and we've had the one in 2023 that was purple. In the pink group we've actually had 6 of our isolates over the last 5 years that have fallen into the pink group. And this really does mirror what our European colleagues are seeing. They're also seeing that the red group dominates the population over in Europe, with just some of the isolates falling into the pink group. And it just really shows just how diverse the red group is, because it has this massive, great big section of all these isolates, all with, you know, slightly different virulence profiles, and it's just a very diverse group. So my wheat yellow rust summary. The varieties generally performed as expected in my adult plant field trials. Many of the 2023-24 RL varieties were resistant to all isolates at adult plant stage. I received 92 samples in 2023. We had 1 isolate with virulence for YR8. No virulence was detected for YR5, 10, 15 and 24. We had 2 common pathotypes. And the genotyping, we had 21 red group isolates, 1 pink group isolate and 1 purple group isolate. So before I move on to the brown rust, has anyone got any questions?
Thanks, Amelia. Just a quick question about your young seeding tests and your scoring. Susceptible, if it's susceptible to all 5 races or just 1 race, they're quite different, aren't they, depending on the frequency of those races out in the field. I mean, if it's resistant to all races, R. If it's susceptible to all races, S.
It's susceptible to just 1 of my 5 isolates.
That's what I'm saying. So if you're only susceptible to 1 and you're still scoring it S.
Yes, we've actually been in discussions with AHDB to see if we need to tweak that R and S that's on the... we are going to look at that. We haven't quite decided how we're going to do it yet. We're thinking maybe is there a way we could do a big S and a small s to say that it's, you know, more isolates were susceptible, you know, or
put a number in front of it.
We could just put a number. We're trying to make it as it.
1S, 2S, out of the 5.
That's a good idea. Please put that on your feedback forms. If you've got anything like that you think, well, actually, it would make more sense if you did this, please let us know. To be quite honest, we look at this data and we get a bit blinded by how relevant it is and how useful it is to you, and that's what we need to know. We want to know what we can do to improve this service, what we can do to make it better for you, make it more user friendly. So anything like that, please put it on your feedback forms.
Just for growers to know that, you know, looking at the data, if you've just got R and S at seeding stage, that's not.
Yeah, we're on the case, but you've come up with a great suggestion there, so we'll take that on board. Yep.
Although what you've presented is for wheat, but do you know if for triticale it's the same races of yellow rust infecting, or is it completely different?
So we don't include triticale as part of the survey, but over the years, when somebody sent me a sample in, I have tested it, and I don't have a triticale diff set, but I test it on my wheat set, and basically it seems to be not a lot different to what I'm seeing with my wheat isolates. So it's the same sort. It will affect the same sort of varieties as a normal wheat isolate. So I don't have any specific information on triticale, but at the moment I think it's wheat's, you know,
That's
infecting
great,
it.
yeah. Thank you.
Wheat yellow rust infecting it. Any more questions?
With the isolates that you find on a particular variety, and then they are not able to reinfect that variety, well, how do you explain that?
So we actually see that quite a lot. I see it at seeding stage. So I get let's say a sample in from Castello. It doesn't infect Castello at seedling stage. We get an isolate in from Caddo Siskin or something. Doesn't infect Siskin at seedling stage. So some of them, some of the varieties I do include in my normal seedling differential sets. Like we don't mention it, but I've included siskin in there for a number of years just because we were interested in the resistance that siskin carried. And a couple of years ago, we were seeing isolates coming in from siskin. So I wanted to include that in my pathotyping. But yeah, I can't explain it. It could be that it's particular sort of like environmental factors out in the field that's making it more, you know, more aggressive or more, you know, more likely to infect that variety. The same with my adult plant trials. I don't see, like my Costello and Mayflower isolates didn't infect Costello and Mayflower at adult plant stage. So we do see it. I can't explain it, but it could be. We don't actually have any way of verifying the host variety of the sample that's come in, and it's another thing that we're discussing with AHDB as to way we could get that verification done, so we can, if we have a sample in from a highly resistant variety, we could get that perhaps genotype, the variety, you know, and so we are looking at that at the moment. All I'm doing is relying on the information that the sampler has sent in. So if you are sending in samples, please give as much detail as you can on the sampling sheet. And also, if you take a sample and you think it's from variety X, and a couple of weeks later you or the farmer realise that it's not variety X at all, please tell me. It doesn't actually make any extra work for me, because all I have to do is change it on my little database and just say, originally thought to be, you know, variety X, it's actually been now confirmed as variety Y. So if you, and I do get that, you know, not that often, but you know, now and again we'll have somebody just drop us an email just to say, oh, actually I sent you a sample last month. It's not the variety that I thought it was.
Do you ask for details of what was it? Foci in the field, was it a single plant? Do you get that information?
We do get in that information. Charlotte's got our actual sample sheet picture, so you can see we ask for, if it's for trial ID as well as location. We ask for growth stage. We ask for percentage plot infection, or sometimes somebody might just say, oh, scattered leaves across the field, or you know, several foci. It's whatever information we samplers provide, we record that and we make a note of that.
So on that same topic, so you're saying that most of the samples you receive, they're on adult plant leaf material?
Yes. So we do get, sometimes I do get samples in the autumn, so obviously that's on very young plants that have, you know, been sown in the autumn. But we only get a couple of samples in. I haven't had any in this year yet. But a lot of the samples we get in kind of start when the trials managers are starting to make their field assessments and walking their trials. So March onwards is generally the time we start seeing samples. I sometimes get samples in January and February, but not so
that would
often.
have an effect on results. It's come off an adult plant and you then do a seedling test. It's very likely that sickness, it could be susceptible or even or resistant, could change the phenotype.
Yep. So you know, I record that all when the sample comes in, what growth stage it was, and we do try and always test samples from the beginning of the season and at the end of the season. So usually we will test, like I tested 23001. That one came in January last year, and I tested 2392, which was the last one in June.
So just on the seedling testing, you still test on all the differentials, and it seems to me that for a lot of the virulence, like 1, 2, 3, they're pretty fixed in the pathogen population. So maybe you can think about not testing on those, just test on 8, 5, and then that would give you more room to test some of the recommended list candidates.
That's exactly what we did in 2023. So me and Charlotte whittled down. So for what we were doing in 2022, we had this differential set that had 48 varieties. And some of the differentials maybe had duplicates where you had 2 different differentials for the same resistance gene. So we cut all the duplicates out. We actually cut out some of the additional cultivars, which we then realised that actually some of them were quite key different additional cultivars. But We cut them out so we could include more RL and RL candidates. And I don't want to mention it too much because poor Charlotte's going to mention it in her slides, but we're now going to do a change in 2024. But I'll let Charlotte talk.
I mean, Virulence to 1, 2 and 3 have been around since I
Yeah, no, to be quite honest,
that's a long time.
we have this database that dates back to the year dot, and when you try and compare pathotypes over the years, if you cut out them, you cannot compare it across years. But we've decided that actually we want to know what's happening now. The RL varieties is they're the hot topic. That's what we want to include. So we will be cutting out YR1, 2, maybe 3 and 4 do change a little bit, and there's some isolates that are detected that don't that are aberrant on YR1. So if we cut that one out, we wouldn't know if that was detected or not, but we will be definitely looking at that in 2024, and I'll let Charlotte talk about that.
I think we just need to move on in terms of.
Oh yeah, because I've got the brown rust to go. Yeah.
Just hold on to it because we might have a little bit of time at the end, or you can come and find us during the break.
Right. So, any questions? Sorry, everyone. So now we have the wheat brown rust. I'm just going to take a bit of water before my throat dries up. Right, I'm ready. So for the wheat brown rust, so we do exactly the same work with the wheat brown rust. The slides will be very similar, but in the top right-hand corner, I've now put WBR. Before we had WYR in the top right-hand corner. And I've cut out some of the slides, so it won't be quite as many slides as the yellow rust. But we're going to start with the adult plant trials for wheat brown rust. So these are the isolates that we chose to infect those 5 trials. We selected 22006 from Cambridgeshire. That one came in from Siskin, and it was selected because it had this pathotype that carried virulence for LR2C. So with yellow rust, I was always talking about YR genes. With brown rust, I'm now going to be talking about LR genes because brown rust's other name is leaf rust, so that's where the LR comes from. We also selected 22014, another one from Cambridgeshire. It came from Crusoe, and this one had a new pathotype. In 2023, we selected this one, 22118 from Oxford. That one was selected because it was the second most common pathotype. We selected 22024 from Theodore in Dorset. That one was particularly interesting because it had virulence for LR24, and that particular isolate has now been chosen and is being used to inoculate RL, VL adult plant trials. So it's basically, it was selected, but we hadn't tested it as an individual isolate at adult plant stage. But it's as you'll see, it's been a good choice for as a RL-VL isolate, as I'll explain in a minute. We also selected 22026 from Mayflower, and that one was selected because it was the most common pathotype in 2022. So again, I've expressed the adult plant trial results in terms of a mean percentage plot infection. This time it's over 7 assessments from growth stage 43 to 77. Again, each isolate is represented by a different coloured bar. So we've got 22006 in the blue colour, 22014 in orange, and so on. So first up we have Buster, and that's our most susceptible control variety. It's one we usually include in adult plant trials. And it was all isolates infected Buster. So Crusoe, with a rating of 3, was our most susceptible RL variety in our brown rust trials. You can see it did have higher levels of infection with isolate 22014. Kedewasquium, again, another one rated 3, again susceptible with all 5 trials. And last on the list is SY in situ, rated 6, and again had infection with all 5 trials. I would like to point out the blue line, as it's a little bit lower than the others. I did find that they were all inoculated at the same time, all assessed on the same days, but the infection in the trial with 22006 was very slow to get going. The plants looked quite healthy. I did speak to my RL trials manager, or my trials manager down on this site, and just ask if they knew of any reason why that particular trial was looking quite healthy, and they didn't know if anything had occurred. But it carried on throughout the season, where I just had these lower levels on that particular isolate. So I also have done the same as I did for the yellow rust. I popped up the varieties that are all rated 7 or more. Again, we've had to change the y-axes to illustrate these results to you. So if we start with KWS Sciat with a rating of 7, you can see it was susceptible to all 5 isolates with quite nice infection levels, with the 22024 isolate in brown. RGT Wolverine, again, susceptible with all 5 isolates. Merit with a rating of 7 and LG Prince with a rating of 7 showed very similar disease levels to each other, and saw higher levels of infection with 22014 and 2218. We have KWS Dorsum and LG Astronomer. So Dorsum was infected with all 5 isolates, but you can see it's under 5% mean percent. And LG Astronomer, that one did quite well. It had these slightly higher levels with 22014 and 2218, but still performed well. And Skyfall was my most resistant variety in trials. And you may be thinking, how can she assess Skyfall for brown rust when it's pretty susceptible to yellow rust? I was actually, and I've been lucky for the last couple of years, that the yellow rust in this particular field, it was quite late coming in the season, or in these trials. Yellow rust just wasn't a problem for me last year when I was scoring my brown rust trials, and I was able to score Skyfall quite comfortably right up until that last assessment. So what you see is a true reflection of how it performed in my trials. So I wanted to have a look at that LR24 isolate WBR22024. So you can see I have these lovely high infection levels with Warrior and Theodore. So Warrior is a additional cultivar that we use in our seedling tests and the adult plant trials, which we think carries virulent or the resistance of LR24. So that isolate performed as expected. The same with Theodore. The more I look at Theodore, the more I include it in our trials, it does seem like it carries the resistance of LR24. We don't know that for certain. We don't have that information from the breeder, but it always performs very similar to my other LR24 diffs. And I've just popped up RGT Illustrious, RGT Stokes and KWS Palladium, purely because they were showing slightly elevated levels with 22024 as well. So the candidates, how did they perform? You can see we have lots of brown blocks there, so they were showing these slightly higher levels of infection with brown rust. Not surprising really, because they had sort of like moderate disease resistance ratings of 5 and 6. It was quite interesting to note that Almara did well with 2 of the isolates, where it was 0.04 across those 7 assessments. And also KWS Gatum did quite well in the trial infected with 22026 there, with just a 0.1% mean. And I can't help but hide it, but you can see that this trial here had just overall those lower levels. So my take-home messages for the brown rust adult plant trials: the most susceptible RL variety in trials was Crusoe with a rating of 3. The most resistant RL variety was Skyfall, so it lived up to its resistance rating of 9. LG Astronomer and KWS Dorsum, I think they also did fairly well compared to the other RL varieties. LR24 isolate, WBR22024, infected Warrior and Theodore at adult plant stage. And the candidate variety Almara showed no infection of 0.0% mean with 2 of the isolates when tested at adult plant stage. So now we come on to my 2023 brown rust samples received. So I received 15 samples. They came from 7 counties. So we've got quite, we had some samples in from the southwest and from some of the eastern counties. They came from 10 varieties. So we, Crusoe was actually the most sampled variety. We had 4 out of the 15 samples from Crusoe. Theodore, I had 3 out of the 15 samples. Champion, rated 5, I had 2 samples from Champion. And I just wanted to point out that Theodore, so in 2022 we had that isolate 22024 that was coming from the southwest. But in 2023, again, Theodore was infected, but it wasn't just confined to the southwest this time. Yes, we had Dorset and Devon represented in the samples we received, but also we had a one from East Yorkshire as well. So my brown rust seedling test, like I said, we're funded to test 50 yellow rust isolates and brown rust isolates in total. So I actually tested all 15 brown isolates, and yet that means I tested an extra one of yellow rust that we didn't need to, because I could have got away with 35, but I get a bit carried away and end up testing, you know, an extra one. So these are the isolates that we selected for, that we tested. So we have a range of host varieties. We've got Theodore in there, we've got Crusoe in there, Mayflower, and the locations. So yeah, we have we tested one from all of the samples that came in. So now I'm not going to show you all of the resistance genes for brown rust. I've just picked out some of them here. And so these are the results on my brown rust seedling differential tests showing the percentage of isolates carrying virulence. And again, I've expressed it for the last 5 years. And you can see virulence for LR2C, that one has declined. It wasn't detected in my 2023 samples. Virulence for LR24 has increased. So you can see it's gone from 12% in 2022 and it's kind of shot up to 40% in 2023. Before 2022, we hadn't seen virulence for LR24 since about 2017. So that's definitely a change in the brown rust population there. And virulence for LR28, that one plateaued out at 60%. And it was the same as 2022 levels. No virulence was detected for LR2A, LR2B, or as we've seen, LR2C. So I just wanted to have a look at the additional cultivars. So Stig and Warrior are both thought to carry LR24, and I popped Theodore in there. I've put a question mark by it because we don't know for certain, but it does seem to have perform very similar to my LR24 differentials. And I've also just popped up Robagus and Firefly there because they are the additional cultivars that we use for LR28. And again, they perform very similar to each other. So the pathotypes that we found in 2023, we had this common pathotype at the top there. And then we have the second most common pathotype with 20% frequency had LR24. So that's a change, that's an increase from last year. The new pathotypes that we identified, there were 5 new pathotypes, and you can see 3 of them carried virulence for LR24. So yeah, we're definitely seeing that increase for virulence for LR24 from the previous year. So again, for the this piloted screen that we did on the RL subset, so again, my 15 isolates were tested on a subset of the RL varieties. Again, we picked out ones that we thought would be interesting to have a look at against those 15 isolates. So in this screen, all RL varieties tested were susceptible to 5 or more isolates at the seedling stage. So there's most varieties on the recommended list, or all varieties on the recommended list, are susceptible at seedling stage to brown rust. But again, for the brown rust, we've taken just 5 of those isolates and tested it on the full set of RL and RL candidates. And these tests I've literally only just completed. They were in the growth rooms over Christmas. I scored them on the first week back over Christmas. So this is brand new results. It hasn't been published on the recommended list yet. But this is what I found, is that they were all susceptible to 1 or more of those 5 isolates. But the keen-eyed amongst you will notice there's actually one missing from my slide here. So all of those susceptible. I had 1 variety was resistant to all 5 isolates at when I tested them on the full set and the RL and the RL candidates. And it's RGT Goldfinch, which is one of our candidate varieties for this year. So, and it was really interesting to see that Goldfinch, it didn't have any pustules, it didn't have any, no brownness whatsoever. And I was very pleased with these tests. They all worked well. Goldfinch did really well. So my brown rust summary. The varieties performed as expected in adult plant trials based on their RL rating. We received 15 samples in 2023. 100% of the isolates carried virulence. I didn't put this on the slide because the slides were getting a bit busy, but I'll put it in my summary points that 100% of the isolates carried virulence for LR1, 10, 13, 14A, 15, 16, 17 and 37. Virulence for LR24, as we saw, has increased to 40%. We detected this particularly prevalent pathotype in our 2023 samples. 5 new pathotypes were identified in 2023, and all but one of the RL and the RL candidates were susceptible at seedling stage, and it's that candidate variety, RGT Goldfinch, which was resistant at seedling stage to 5 isolates from the 2023 survey. I'm hoping I now have a slide with questions. So any questions on the brown rust? Hi, Kostya.
How this variety is scored in the adult test?
Pardon?
How this Goldfinch variety is scored in the adult plant test?
So it's brand new candidate variety, so it actually hasn't been tested at adult plant stage yet. It would have been tested in 2024. But Charlotte's got some more information on that next.
In terms of the quality samples you receive, is there any preference or standard?
Sorry, I've got
The quality of the samples?
the quality.
Yeah.
So the quality of the samples we get for both yellow rust and brown rust do vary depending on how quickly the sample has managed to get it in the post, how long it's been in the post. So we have this free post address that Charlotte will mention. So sometimes it can arrive to me within 3 days, sometimes it can take 5 days. Hopefully won't get any Royal Mail strikes this year, because they do mean the sample can deteriorate the longer it is in the post. So basically, I try my hardest with these samples, and for the brown rust, I managed to get every single one working. For the yellow rust, I think there might have been either between, I think it was 2 that failed outright. I couldn't get them going. But on a normal sample year for yellow rust, when I get about 100, maybe say 120, 150, my success rate is usually about 5 per year will fail. And so what happens when a sample, I've got to be quick because otherwise Charlotte won't have much time. When a sampler sends me their samples, I first incubate it, and the reason we ask for you to send several leaves in is because although I only need Like for yellow rust, I only take one stripe. For brown rust, I take one pustule. I like to choose that stripe. I like to choose that pustule. So I'll incubate quite a few leaves, and then after a couple of days, I can actually select and go, ooh, that one's looking good. I'll have that one. So usually I'm quite, I don't want to sound big-headed, but I am quite successful in getting my isolates going. But I think that's just years of experience. Any other questions? Hi, Wesley.
So when you put up your percentage infection on your adult plant testing, and then you have the variety with its rating of 1 to 9,
Yep.
it would be nice to have the percentage infection converted to the 1 to 9 scale, so you can see, you know, if you've got a variety that's got a scale of 8 and it's got 10% infection, does that mean it's now a 7 or a 6? Or?
There isn't actually a cut-off point for the different. There isn't a key or scale that I can say.
To do that conversion, because it's a bit difficult to relate what that level of percentage infection means to the rating of that variety.
Yeah, so currently it would have to be a NIAB, key. There isn't a standard key that you can just say a rate, that rating of 9 should have no or under X percent.
Thing, but yeah, it's just for understanding of the data for people that aren't, expert pathologists.
Yeah, so to be quite honest, we try our best to present the information so people can understand it both ways. If you're a grower, you'll understand the RL rating, so you can see that's like Skyfall, low rating, and it is susceptible. But yeah, we can certainly have discussions about that and see if there's a way we can do that in the future for you. You had a question?
I'll just look at, maybe my eyes, but the goldfinch looked a bit yellow. I was just wondering if there was any physiological
On my little
impacts from the
picture?
plant defences.
On the picture?
Yeah.
That I didn't actually. It's just a picture from our archives. It's not
So
actually
no
RGT
physiological
goldfinch
impacts.
in that picture.
Okay.
It's just for demonstration
Just wondering about the plant
purposes.
defence. Okay, thanks.
I must admit, I was kicking myself that I didn't take a photo of it. And I'd done the test, went back up to my computer, but inputted the results and thought, oh, RGT goldfinch. And I thought, oh, I'll go back to the growth rooms, take a photo. But because I'd left it a couple of days, I thought I can't take a photo of that because they go over quite quickly in our growth rooms. The temperature is quite high, the brown rust. Because the plant carries on growing, the first leaves actually sort of like die and wither off after a period of time. And I thought, I can't take a photo of it now. It doesn't look very happy. But when I did assess it, because I assessed them on for brown rust, for yellow rust, I scored them twice on day 14, and then again I usually just check them on day 18 for the brown rust. Because it's a warmer temperature, I like to score them on about day 11, and then I do another day 14 score. So you get 2 scores out of them, but quite quickly after that, all the varieties would tend to deteriorate because it's the second leaves and the third leaves coming up, and they're taking all the nutrients. Any more questions? Great, so I'm going to hand you back over to Charlotte now, who'll take you through the final slides. Thank you.
Okay, so I'm just going to talk through some of the improvements that Amelia has already mentioned and some of our plans for 2024. So we're really trying to deliver a faster service, and we want to deliver in-season reporting. So last year, in 2023, we managed to get some bulletins out on of in-season sample data updates. So this included heat maps to show the areas where we were getting samples in from. So we released these on X and on AHDB's website. So please do keep an eye out for these next year. We're hoping that we can include some more information, but it's a work in progress. And so, as Amelia's mentioned, so I'd said at the beginning about what we did sort of traditionally in the survey, and then Amelia's mentioned some of the improvements that we've made. So in 2023, we did screen a larger number of isolates on a subset of the RL varieties. However, because we'd only done it on a subset, we weren't able to put this information into the RL booklet. So from now on, we're going to be looking at all of the RL varieties and candidates so that this information can be included in the RL booklet. So we're looking at doing more than 5 isolates And we want these in-season tests to be completed earlier, and also for the data to be released so it's ready for that first edition of the following year's RL booklet. So the data needs to be ready by about November. So whilst we did have some information ready in 2023, we didn't have enough and all of the right information. So it was a step towards what we want to do last season. And as you will have noticed, we're holding an earlier stakeholders meeting, and we want to disseminate the information earlier. So we want to get all this information out in the winter so that the agronomists and growers have this information much earlier than they have previously. I just wanted to talk a little bit about the AHDB watch list. So these were introduced for yellow rust in 2021 and for brown rust last year. So these lists, they flag unusual levels of disease observed in some trials, and this is compared to the average across all trials. So this extra layer of information provides a valuable warning of any potential change in the UK's rust race structure. And it orders varieties based on their disease levels in the 3 worst trials of that year. And it ranks them from highly resistant, medium resistant to low resistant. And the new list for 2024-25 is due to be released very soon. So these are usually released updated in January or February. I've had a look at the data, and most varieties perform in line with their main RL rating. However, we do have some varieties that have been flagged. So I do have to add a caveat that for yellow rust, there was very limited data for 2023 from the natural infection trials. But these varieties that are listed on the screen performed slightly worse than their RL rating indicated. So ones to just keep an eye on this season, and to see if the fact that it was limited data in 2023 made a difference. Perhaps they'll perform better or in line with their RL rating this season. And then for brown rust, we've got Theodore, which was flagged in 2022. Again, still performing worse in some trials. And probably because of this, these isolates that carry the LR24 gene are more prevalent across the country. So plans for 2024. So we've Amelia's alluded to it, but we're not going to be performing the adult plant trials this year. This is partly due to budgetary constraints, but we're also going to try this year to see if we don't perform these adult plant trials, how does that impact the survey? Can we use data from the RL trials that are out there across the country? So we will see how that goes this year. And as we've mentioned, we want to test more isolates on the full set of the RL and the RL candidates at the seedling stage. So we're not quite sure how many we'll be able to manage, because each season it varies. So last season, Amelia was able to do these additional tests because she only received 92 samples. But in a very heavy year, we're just not able to... We'll see how many we can get through. So it's a work in progress and something that we're improving. And as I've mentioned, we really want to get not only the tests done, but the reporting in season. So these are the things that we'll be focusing on over the coming year. So for wheat and barley powdery mildew, we didn't conduct a survey for these pathogens in 2023. There were no reports of unusual sightings to indicate any major changes in the wheat powdery mildew population. However, the AHDB did have some reports of some susceptibility seen in spring barley, MLO-resistant varieties in Scotland. So in 2024, the UKCPVS is going to look at a small selection of the barley powdery mildew isolates. If we get the same issue this year, then we will test those to see what's going on in those isolates. So I just want to go through the sampling information. So, I mean, we couldn't do the survey without everybody who sends in samples. So thank you to everybody who takes the time to send us in a sample. We really appreciate every single one that's sent in. And every sample is important to us. As Amelia has explained, you know, she does her very best to recover each one of the isolates, and she is an expert and has magic fingers, with only a few escaping her touch. So, yeah, we do make every effort for every sample. They're all treated the same to aid successful spore multiplication. And just to highlight some of the ways that the samples are used, so obviously we run the survey, we provide information, we characterise the isolates, but ultimately what we do with that information is we use the information to select isolates that are on the inoculated recommended list and variety list trials, and these produce the disease ratings for the recommended list, but they also are used to inform which varieties pass through the variety listing process as well. We've mentioned that the UKCPVS seedling and the young plant, so yeah, seedling / young plant data is included in the RL list now to give another layer of information. The isolates are used by some of the breeders to screen their breeding lines, so this is future proofing for commercial resistant varieties. Some isolates go to AgChem and biocontrol research, so trialling new products. And then also some of the isolates go into research studies, so that's both PhD studies, ongoing collaborations with other research institutes, and these are all focused on, or some of them are focused on understanding the complexities of the pathogen at the molecular level, something that we can't do in the survey ourselves, but that other people have the funding and the capabilities of investigating these things. And we have participated in European projects, so without the samples from the UK we wouldn't have such a good resource and valuable input, and we still speak to our European collaborators even though those projects have finished. And as I mentioned earlier, all of the samples go into long-term storage, so if in the future anybody wants to look back at what was happening over the years, have any research question, they can go back and have a look at that snapshot from the season, from the locations around the country. And this year, this is not on wheat, but we're putting a call out for if you see any oak crown rust when you're walking trials or anything, please could you send us in some new isolates. We want to update our isolates that we use for the RL inoculated trials. So you can send them in on the free post UKCPVS address. So if you remember later in the season and you spot any oak crown rust, please do send it in. Amelia mentioned earlier that this is our sampling sheet. So she mentioned all of the bits of information that we request. And there's a link at the top for finding this sampling sheet. The more information you give us, the better, the more we're able to understand about the isolates that are sent in. So please, anything that you think is relevant, please do note that down on the sheet. And please place samples into a paper envelope. Please don't use plastic bags. Amelia's mentioned the free post address, and if you wanted to send it with a stamp for first class delivery or next day delivery, then that's the full address on the slide to come here to NIAB Park Farm. So I just want to go over some overarching take-home messages. So for yellow rust, as Amelia has explained, we didn't see any major varietal breakdowns, but the population still remains diverse. We had the majority of samples coming in from KWX days into the survey in 2023, and one isolate carried virulence for YR8. And as Amelia has explained, the red group dominates the population. For brown rust, virulence for LR24 rose to 40%, and as she mentioned, that candidate variety RGT Goldfinch is resistant to the 5 isolates that we tested at the seedling stage. It'll be interesting to see how that variety performs this year when we were able to test a few more isolates on it. Will it stand up to all the additional isolates? We'll let you know. So please refer to the AHDB RL booklet for the resistance ratings, so both young plant and adult. And also please use the AHDB watch list when making your variety choices. Have the confidence to reduce fungicide use on resistant varieties. Pathogen populations are dynamic and diverse, so please do keep an eye on all varieties and spray if you need to. And as ever, monitor any unusual sightings and send us in a sample if you spot anything. So for further information, there are our annual reports and any bulletins as well that you can find on the AHDB website. The recommended lists and press releases are on the main AHDB website. The new booklet's just been released online, and I believe the paper copies are available from tomorrow. And then there's also information about the sampling and the postage and packaging on the NIAB website. And more information about what's going on globally can be found on the Global Rust Reference Centre. These slides will be made available online, so you'll be able to copy and paste the links. And I mean, the biggest thanks I need to give are to Amelia, who lives and breathes this survey, and she does an absolutely excellent job. But there are also other people that help support the work, both here at NIAB, the samplers that send in samples, the funding bodies and collaborators, Diane and her team. So thank you very much for your attention today.